I want you to stop whatever you are doing right now, just for a second. I want to invite your mind into a completely different, frankly, mind-bending reality. If you can, look out a window. Find a tree, a bush, or even just a tiny blade of grass pushing its way out of a crack in the sidewalk. You are looking at nature's ultimate alchemists.
They really are.
Right. Think about it. These organisms are standing completely still, yet they are actively pulling invisible gases out of the thin air, and they're literally dissolving solid rocks from the ground beneath them, all just to build the physical architecture of life. I've been poring over the source material we gathered for today, and I have to admit, I had a massive breakthrough. It fundamentally changed how I look at a forest or even just a houseplant. We are going on a mission today to understand the exact microscopic blueprint of plant survival.
That is the perfect way to frame it. And for you listening, whether you were just endlessly curious about how the natural world operates at a molecular level, or you are a student specifically sitting down to master this material for a massive high-stakes exam like the Neet, this deep dive is designed to be your absolute masterclass. What we're really talking about today, the core essence of this entire topic, is mineral nutrition. We are looking at the exact chemical roster, the precise atomic blueprint that plants use to survive, to grow, and to reproduce.
Right, and here's where it gets really interesting for me. Because to understand why mineral nutrition is even a topic we need to study, we have to understand the fundamental, real-world biological problem that plants are forced to solve every single day. I was trying to conceptualize this, and I thought of it like this. Imagine you are a construction worker. Your job is to build a massive, towering skyscraper. You need steel, you need glass, you need concrete. But there is a massive catch.
Yeah.
You are super glued to the floor. You cannot take a single step to go fetch your materials. You literally have to stand there reaching your hands out and wait for the wind to randomly blow a steel beam into your grip. You have to wait for the rain to wash liquid concrete over your boots so you can soak it up. That nightmare logistical scenario. That is the exact plight of the plant.
It is a profound logistical nightmare. And it answers the question of why this field of biology exists in the first place. It exists because it solves that exact problem of resource acquisition. Every living thing, whether it's you, me, or an oak tree, needs to build DNA, construct proteins, and fortify cell membranes and cell walls. Those macromolecules aren't magic. They require very specific atomic building blocks. We are talking about atoms of nitrogen, phosphorus, calcium, magnesium. But the plant must extract these essential building blocks from a highly variable, unpredictable, and sometimes hostile environment, namely the dark soil and the open air.
Exactly. And so for our mission today, we are going to build a clear mental map of how this all works. We are going to trace the history of how scientists actually figured out what plants eat, so to speak. We'll look at the exact chemical VIP list of elements they need. Then we are going to play plant doctor and learn how to diagnose diseases when things go wrong in the soil. And finally, we're going to unpack what I honestly believe is a biological miracle, the absolute bottleneck of Earth's ecosystem, nitrogen fixation. So let's go back in time for a second, because before we could understand the chemistry, we had to invent a way to observe it. I was reading about this German botanist, Julius von Sachs, back in 1860.
Julius von Sachs. If you were tracking the history of botany, 1860 is a watershed moment. Before Sachs, if you asked anyone, even scientists, how plants grew, they just assumed soil was this magical, complex black box that plants absolutely needed to survive. Soil was thought to be the irreplaceable life force.
But Sachs completely shattered that assumption, right? He demonstrated for the very first time that plants could be grown to full maturity from a seed all the way to producing their own seeds in a completely defined nutrient solution in the total absence of soil. He essentially invented what we know today as hydroponics. I honestly look at hydroponics as a 19th century biological hack. He hacked the system to isolate the variables.
Calling it a hack is highly accurate in terms of the scientific method. The essence of Sachs' method, and all the vastly improved hydroponic methods developed since 1860, involves culturing plants in a soil-free, highly defined mineral solution. But to do this successfully, and to actually learn anything valid from the experiment, our sources emphasize that the methods require strictly purified water and strictly purified mineral nutrient salts.
Okay, wait, slow down for a second on that point. Why the intense focus on purification? I mean, plants drink muddy rainwater in the wild all the time. Why does it have to be perfectly purified water in the lab?
It's a great question, and it connects directly to how we design experiments. If we connect this to the bigger picture of the scientific method, if your water isn't pure, you are introducing unknown variables into your observation. Imagine you were trying to figure out if a plant needs zinc to survive. You mix up a solution with 0 zinc, but you use tap water. What if your tap water ran through old pipes and picked up microscopic trace amounts of zinc?
Oh, I see. The plant would survive, and you would write down in your notebook, plants do not need zinc. Completely unaware that the tap water secretly fed it to them, you'd ruin the entire experiment.
Exactly. You need an absolute blank canvas. You start with purified water, essentially just H2O and nothing else, and you add elements one by one, watching what happens to the plant. And the crazy thing they discovered when they started doing this, analyzing what plants actually absorb from the wild, is that plants are absolute hoarders.
They really are. As analytical techniques improved, scientists started finding things inside plant tissues that made no sense. Out of the 105 or so elements discovered so far in the periodic table, more than 60 can be found inside various plants.
Yes, and that brings up some wild examples.
The sources pointed them out. There are plant species that accumulate selenium, there are plants that literally accumulate gold from the soil, and most shockingly, plants growing near nuclear test sites will actively take up radioactive strontium.
Which brings us to a massive high-yield concept, especially if you are facing down an examiner in a testing hall. This is a classic trap. Just because an element is found inside a plant like gold or radioactive strontium does not mean it is essential for the plant's survival.
Right, the plant isn't using gold to build its cells, it just accidentally drank it up because it was in the water.
Precisely. Examiners absolutely love to test the difference between mere absorption and true essentiality. Plants will absorb all sorts of things passively, almost like a sponge soaking up dirty water. But to define the molecular blueprint of a plant, we need strict rules to define what they actually need versus what they just happen to absorb.
This felt like reading the entry requirements for the strictest VIP club in the biological world. The scientists established the criteria for essentiality. There are 3 golden rules an element must pass to be considered truly essential.
Let's break those down because this is foundational. Here is what you must remember. The 3 golden rules of essentiality. Rule number 1. The element must be absolutely necessary for supporting normal growth and reproduction.
So if that specific element is missing, the plant simply cannot complete its life cycle. It won't grow properly, or it won't be able to set its seeds for the next generation. It's a hard start.
Exactly. A hard stop to the life cycle. Rule number 2. The requirement for that element must be highly specific, and it cannot be replaced by a stand-in element.
I really like this rule because it shows how precise the biochemistry is. If a plant is dying because it lacks, say, magnesium, you can't just look at the periodic table, find another metal like calcium, throw extra calcium into the dirt, and expect the plant to adapt. The deficiency of one element cannot be met by supplying a different one. It's magnesium or death.
It is biochemically non-negotiable. And finally, rule number 3. The element must be directly involved in the metabolism of the plant.
Meaning it has to be an active participant in the chemical engine. It's actually part of a molecule doing work or structural support, not just floating around as a bystander. Life cycle, irreplaceable, metabolic involvement. It's so elegant.
It is. Now, before we move on to the actual roster of elements that pass this strict test, I want to briefly touch on how far Sachs's hack has come. Hydroponics isn't just a 19th century scientific experiment anymore. It has transitioned into a massive modern industry.
Yeah, the sources mention it's successfully employed for the commercial production of major crops like tomatoes, seedless cucumbers, and lettuce. Which makes sense. If you know exactly what the plant needs, why bother with the unpredictability of soil? You just give them the perfect IV drip of nutrients.
It is phenomenal for yield. But there is a massive caveat that early commercial growers learn the hard way, and it's something you must understand about root physiology. The nutrient solutions must be adequately aerated to obtain optimum growth.
Wait, aerated? As in bubbling air into the water? I thought roots just needed water and minerals. Why do they need air if they are underground anyway?
Think about what roots are made of. They are living cells. And what do living plant cells need to do to generate energy? They need to perform aerobic respiration, just like our cells. If a hydroponic solution is stagnant and poorly aerated, the dissolved oxygen depletes rapidly. The roots literally suffocate and drown. They need oxygen to generate the ATP, the cellular energy, which, as we will see later, is absolutely required to actively pump those mineral nutrients into the root against a concentration gradient.
Oh, wow. So if they can't breathe, they can't eat. That explains why in those commercial hydroponic setups, you always see a pump circulating the solution, usually flowing down a slight incline to mix with the air. That movement is vital.
Exactly. So we know how to test for essentiality, and we know they need an aerated environment to drink up the nutrients. What exactly made the final cut? Who is in the VIP club of elements?
Based on those three strict criteria, only a relatively few elements have been found to be absolutely essential for plant growth and metabolism. And scientists divide them into two broad categories based entirely on their quantitative requirement. Basically, how much does the plant actually need to build its skyscraper?
This brings us to the quantitative divide. Macronutrients versus micronutrients. And there's a very specific, defined threshold separating them.
The sources define macronutrients as elements that are generally present in plant tissues in large amounts, and there's a specific mathematical threshold given. They must be present in excess of 10 millimoles per kilogram of dry matter.
Let me pause you right there, because for our student listeners, this is a high-yield threshold. Examiners love testing precise definitions. You must memorize that exact number and unit, 10 millimoles per kilogram of dry matter. If the plant's requirement is in excess of that, it's a macro. If it's less than that, it's a micro, which is also commonly called a trace element.
Okay, 10 millimoles per kilogram. But I have a clarifying question here. The text specifically says dry matter. Why dry matter? Why not just weigh the plant fresh out of the ground?
That is an excellent clarifying question. Think about a fresh, crisp lettuce leaf. What is mostly inside it?
Water, like 90% water.
Exactly. And the water content of a plant fluctuates wildly depending on the time of day, how much it rained, and the humidity. If you measure the elements against the fresh weight, your percentages will be totally skewed by the water weight. So scientists take the plant tissue and literally bake it in an oven at a low temperature until all the water completely evaporates. What is left behind is the dry matter, the actual structural mass of the plant. Measuring against the dry matter gives you an accurate, stable baseline for how much of an element is truly integrated into the plant's body.
That makes total sense. Remove the fluctuating water variable. Okay, so looking at the dry matter, let's look at the macros, the big builders. There are 9 of them. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium.
Now, we should build a bridge here to what you might already know from basic chemistry or biology. The first 3, carbon, hydrogen, and oxygen, the classic C, H, and O, are somewhat special cases. They are mainly obtained from carbon dioxide in the air and water from the soil. They are the structural backbones of all organic chemistry, carbohydrates, lipids, proteins.
Right, they are the literal mass of the plant. And then the rest of the macros, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, these are the ones primarily absorbed from the soil as mineral nutrition.
Correct. 9 macros in total. And then we have the micronutrients, or trace elements. These are needed in very small amounts, less than 10 millimoles per kilogram of dry matter. There are 8 of these: iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel.
Iron, manganese, copper, molybdenum, zinc, boron, chlorine, nickel, 9 macros, 8 micros. That's 17 essential elements in total.
And a common mistake students make is simply forgetting which elements belong to which list. You need to have these 2 lists locked in your mind. If you see zinc on a multiple choice test, you must instantly recognize it as a micro. But wait, nature always throws in a little extra nuance.
Oh, right, the beneficial elements. The sources note that in addition to these 17 absolute essentials, higher plants, like advanced crop species, specifically require some beneficial elements. The text lists sodium, silicon, cobalt, and selenium. They might not meet the strict 3-rule essentiality test for every plant on Earth, but for many higher plants, they are highly beneficial for optimal growth.
That's right. So we have our quantitative categories based on weight. But what I find even more fascinating is how we can group these 17 essential elements functionally. What is their actual job in the plant factory? The scientists grouped them into 4 broad functional categories.
This part was amazing to me because it brings the chemistry to life. Category 1. Essential elements as components of biomolecules, making them the structural elements of cells. This is your carbon, hydrogen, oxygen, and nitrogen.
Think of these as the bricks, the steel beams, and the glass of our skyscraper. They literally build the physical body of the cell. They form the cellulose in the cell walls, the phospholipid bilayer of the membranes, and the physical structure of the proteins.
Then we have category 2, and this is where things get really elegant. Essential elements that are components of energy-related chemical compounds in plants.
This is a brilliant evolutionary adaptation.
It really led to my first mind-blowing realization of this deep dive. When you hear the word minerals, you just think of dead rocks in the dirt. But look at magnesium. It isn't just floating around aimlessly inside the leaf. It is perfectly, precisely locked into the very center of the ring structure of the chlorophyll molecule. It's perfectly positioned to catch light energy from the sun. The plant is taking a metal from the dirt and turning it into a solar panel. and phosphorus. It is the literal P in ATP adenosine triphosphate, the fundamental energy currency of all living cells. It's perfect molecular architecture.
Turning dirt into solar panels and batteries. That is a beautiful way to visualize it. Moving to category 3, elements that function to activate or inhibit enzymes.
Right. The factory needs machines, and enzymes are the molecular machines. We just mentioned magnesium in chlorophyll, but Mg2+ has another massive job. It is an activator for ribulose bisphosphate carboxylase oxygenase, which everyone just calls Rubisco, and phosphenol pyruvate carboxylase.
And we cannot understate the importance of that. Rubisco is arguably the most abundant protein on planet Earth. Both of those enzymes are absolutely critical in photosynthetic carbon fixation. Without magnesium activating them, the plant cannot pull carbon out of the air. Photosynthesis grinds to a halt.
And it doesn't stop there. Zinc, or Zn2+-ese, is an activator of alcohol dehydrogenase, and molybdenum is an activator of nitrogenase during nitrogen metabolism, which is going to be the absolute star of our show a bit later.
Finally, category 4, elements that alter the osmotic potential of a cell. This is all about water balance and pressure.
Potassium plays an incredibly important role here, right?
It does. If you recall how stomata, the microscopic pores on the surface of a leaf, open and close to breathe in CO2, it is entirely driven by potassium ions. The plant actively pumps potassium ions into the guard cells surrounding the pore. This lowers the water potential inside the cell, causing water from surrounding tissues to rush in via osmosis. The guard cells swell up, become turgid, and bow outward, opening the pore. To close the pore, the plant just pumps the potassium back out. The element is acting as an osmotic trigger.
It's a beautifully balanced system: structural, energy, enzymatic, osmotic. So we have our broad categories. But what happens when we zoom in on the specific quirky jobs of individual elements? We mentioned a few, but I want to get into the heavy hitters. Let's look at the specific roles of the macros and micros.
This is where we see the intense division of labor in the plant's metabolic factory. This section is essentially a roll call of biological function. Let's start with the undisputed king of the macros. Nitrogen.
Nitrogen. The sources state this is required by plants in the greatest amount of any mineral element.
It is the ultimate builder. It's absorbed mainly as nitrate, NO3, though sometimes plants can take it up as nitrate, NO2, or ammonium, NH4 plus SAC.
And it's needed everywhere. The text specifically highlights that it's required by all parts of a plant, particularly the meristematic tissues, which are the actively dividing growing points at the tips of roots and shoots and all metabolically active cells. Nitrogen is the major constituent of proteins, nucleic acids, vitamins, and hormones.
If carbon is the brick, nitrogen is the scaffolding and the workers. Next is phosphorus, which plants absorb from the soil primarily as phosphate ions, either H2PO4 or HPO42. We already marveled at its role in ATP, but it's also a crucial constituent of cell membranes.
Right, the phospholipid bilayer. Every single cell membrane has a phosphate head, and as part of certain proteins, and naturally all nucleic acids and nucleotides. You can't build DNA or RNA without a sugar-phosphate backbone. The text also mentions it's required for all phosphorylation reactions.
Which means anytime a cell wants to turn a protein on or off by attaching a phosphate group, it needs dietary phosphorus to do it. Next, we have potassium, K+2. We talk about its osmotic role in opening and closing stomatal guard cells, but its resume is much longer than that.
It says it helps maintain an enrichment balance in cells. It's involved in protein synthesis, the activation of enzymes, and it's vital for the maintenance of turgidity of cells.
Let's expand on turgidity because it's crucial. Turgidity is what keeps a non-woody plant standing upright. When a plant lacks water, it wilts and flops over. Potassium ensures that the cells hold enough water to stay plump and rigid against their cell walls, acting like a fully inflated tire.
Got it. Okay, now here is an element I really want to focus on because its physical properties explain so much of plant disease later on. Calcium, Ka2 plus tauret.
Calcium is fascinating. Plants absorb it from the soil in the form of calcium ions. It is intensely required by meristematic and differentiating tissues.
During cell division, it is used in the synthesis of the cell wall, and the sources point out a very specific structure. It forms calcium pectate in the middle lamella.
The middle lamella. Think of two plant cells side by side. They each have their own rigid cell wall. But how do they stick together to form a solid tissue instead of just sliding past each other? They are glued together by a layer called the middle lamella, and calcium pectate is the primary ingredient of that glue.
I think of it like the mortar between two brick walls, locking the adjacent plant cells together.
That is a perfect analogy.
Right.
And because it acts as that structural mortar, once it is laid down, it becomes permanently locked in place. This is a massive concept.
Yeah.
Our sources explicitly note that calcium accumulates in older leaves. Why? Because it cannot be easily broken down and moved around. Once it's built into that wall, it's cemented in.
Wait, so if it's mortar, the plant literally can't tear down the wall to get it back?
Exactly. It's structurally permanent.
That structural permanence is going to be a huge plot point when we talk about nutrient deficiency. But before we move on, calcium has a few other jobs. It's needed during the formation of the mitotic spindle during cell division, so it helps pull the chromosomes apart, and it regulates metabolic activities and membrane functioning.
Moving on to magnesium, Mg2 plus NTM. It is absorbed by plants in the form of divalent magnesium ions. We know it forms the ring structure of chlorophyll, but it also activates the enzymes of respiration and photosynthesis. It's involved in the synthesis of DNA and RNA. And this is a detail examiners love. It helps maintain the physical structure of ribosomes.
Ribosomes are the protein factories of the cell, right? They have a large subunit and a small subunit. So magnesium is the element that literally holds those two pieces together so they can read messenger RNA. That's incredible.
Exactly. And the final macro to highlight is sulfur. Plants obtain sulfur in the form of sulfate ions, SO42.
Sulfur is a structural necessity for proteins, specifically because it's present in two very specific amino acids, cysteine and methionine.
If you are taking notes, highlight cysteine and methionine. The disulfide bonds formed between cysteine molecules are what give proteins their 3D shape. Sulfur is also the main constituent of several coenzymes, vitamins like thiamine and biotin, coenzyme A, and ferredoxin.
Okay, those are the macros, the heavy lifters. Let's shift our focus to the micros, the trace elements, and I want to start with iron, Fa3 plus weak.
Let me jump in here immediately because this is a massive memory anchor for any student. Here is what you must remember. Iron is essentially the bridge between macros and micros.
Wait, what do you mean by bridge? Is it a macro or a micro?
It is technically classified as a micronutrient because it falls below the 10 millimole threshold. However, it is required in the largest amount compared to all the other micronutrients. It straddles the line. Plants obtain iron in the form of ferric ions, Fa3 plus weight.
And its main job is electron transport, right? It says it is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes.
Yes. Think of the electron transport chain in respiration or photosynthesis like a wire carrying electricity. Iron is the conductive metal inside the wire. It's doing a little chemical dance during electron transfer, being reversibly oxidized from Ig2 plus to F3 plus and back again as it passes the electron along.
It also activates catalase enzymes, and interestingly, it is essential for the formation of chlorophyll. Even though magnesium is the atom inside the chlorophyll ring, the plant needs iron to actually build the ring in the first place.
Now for a pair of elements that examiners absolutely constantly test together, manganese, Mn2 plus Ary, and chlorine, Cl.
The water splitters.
Precisely. Manganese is absorbed in the form of manganese ions. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. Chlorine is absorbed as the chloride anion. Along with sodium and potassium, it helps determine solute concentration and anion-cation balance. But their shared claim to fame is water splitting.
Let's really pause and think about what that means. The best defined function of manganese, along with chlorine, is the splitting of water to liberate oxygen during photosynthesis.
The photolysis of water. When sunlight hits the leaf, the plant needs electrons to power photosynthesis. Where does it get them? It rips them out of a water molecule. When you rip the electrons out of H2O, what is left? Oxygen gas. That specific chemical reaction facilitated by manganese and chlorine is the reaction that provides almost all the oxygen in Earth's atmosphere. Every breath you take, you owe to manganese and chlorine. You absolutely must associate those two elements with oxygen evolution.
That is awe-inspiring. Okay, a few more micros to round out the roster. Zinc, Zn2 plus ions, is obtained as zinc ions. It activates various enzymes, especially carboxylases. But the standout fact here is that it's uniquely needed for the synthesis of auxin.
Auxin is a major plant hormone responsible for stem elongation and growth. No zinc, no auxin, the plant becomes incredibly stunted.
Copper, Cu2 plus Ana, is absorbed as cupric ions. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reactions and is reversibly oxidized from Q plus to C2 plus Ana.
And then we have boron, absorbed as BO3 or B4072. Boron has a very specific, almost eclectic resume.
It's required for the uptake and utilization of calcium, membrane functioning, cell elongation, and cell differentiation. But the two functions that really stand out in the text are pollen germination and carbohydrate translocation.
Let's explain why pollen germination is significant. When a pollen grain lands on a flower, it has to grow a massive, long, microscopic tube all the way down the style to reach the ovary. That requires incredibly rapid, directed cell growth and cell wall construction. Boron is the key facilitator for that rapid extension. Without it, the pollen tube fails and the plant cannot reproduce. Examiners love matching boron with pollen germination.
And finally, molybdenum. Plants obtain it in the form of molybdate ions, Mo2 2+. It is a component of several highly specialized enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism.
We will be spending a lot of time with molybdenum when we get to the nitrogen-fixing bacteria.
Okay, so if you are sitting down to take the Neet, expect high-yield matching questions from this entire section. They will give you an element in column A and ask you to match it to its unique function in column B.
Let's do a rapid-fire drill of those associations right now.
Let's do it. Calcium?
Mitotic spindle and middle lamella mortar.
Zinc?
Auxin synthesis.
Boron?
Pollen germination and carbohydrate transport.
Molybdenum?
Nitrogenase enzyme.
And manganese and chlorine?
Splitting water to liberate oxygen.
A perfect summary. It really is a beautifully balanced, microscopic system. Every gear is turning perfectly, but biology is rarely perfect in the real world. What happens to the plant when just one of those gears stops turning? Because the soil ran out of an element, or because there's too much of one?
That brings us to our next major concept. When things go wrong. Deficiency and toxicity. In biology, the absence of a necessity always leaves a signature. Whenever the supply of an essential element becomes limited, plant growth is retarded.
The sources define a very specific threshold here called the critical concentration. It is the exact concentration of the essential element in the plant tissue below which plant growth is retarded.
So if an element is present in the plant below that critical concentration, the element is officially said to be deficient. And because each element has a specific structural or functional job, its absence causes specific morphological changes in the plant's appearance.
We call these morphological changes deficiency symptoms. They are the plant's warning system.
The symptoms vary from element to element, and thankfully, if the farmer or the gardener notices them and supplies the deficient nutrient to the soil, the symptoms disappear. But if the deprivation continues, the plant tissue will eventually die.
Now, here is a fascinating logistical puzzle that I really struggled with at first. The parts of the plant that actually show the deficiency symptoms first, the leaves that turn yellow or die first, depend entirely on the mobility of the element within the plant.
This is one of the most frequently tested concepts in all of plant physiology because it requires you to synthesize structure and function. Let's use an analogy to make it highly intuitive for our learners. Think of the plant not as a single organism, but as a massive manufacturing factory.
Oh, I love the factory analogy. Let's run with that. Let's say the factory is actively building new, state-of-the-art machines in its newest wing at the very top of the building. These represent the young, developing leaves at the shoot apex. But suddenly, there's a supply chain crisis. The roots stop sending up raw materials. The factory runs out of essential elements.
To survive, and to ensure the most critical new structures keep building, the factory manager makes a ruthless decision. They decide to dismantle the old, outdated machines in the oldest wing of the factory, representing the senescent, or older, leaves near the bottom of the plant to scavenge their parts.
It's biological recycling. The plant breaks down the biomolecules in the old leaves and ships the recycled elements up to the young leaves. But UT, and this is the crucial part of the analogy, the manager can only dismantle and move parts that are easily unscrewed.
Exactly. These easily unscrewed parts represent the actively mobilized elements, the highly mobile nutrients within a plant. We are talking primarily about nitrogen, potassium, and magnesium. The plant is incredibly efficient at breaking down old proteins and chlorophyll in the older leaves to extract the N, K, and Mg and shipping them up the stem to the young leaves.
And because the older leaves are having their vital components stripped away, they are the ones that suffer. They show the deficiency symptoms first.
That is the crucial takeaway. For highly mobile elements, N, K, Mg deficiency symptoms appear first in the older senescent tissues. But what about the parts of the old machines that are literally welded to the floor?
The structural components, we talked about this. Elements like calcium, which is cemented into the middle lamella as calcium pectate, and sulfur, which is structurally bound tightly within the physical shape of proteins. These are relatively immobile elements. They are welded to the floor. They are not easily released or transported out of mature organs.
So if the factory runs out of new calcium from the roots, it can't scavenge it from the old leaves. The old leaves get to keep their calcium. The young developing leaves simply have to go without.
Therefore, for immobile elements like calcium and sulfur, the deficiency symptoms appear first in the young tissues.
Let me underscore this for anyone taking an exam. This is a classic trap. Students frequently mix up the mobility rule because it feels slightly counterintuitive. You might think young leaves are weaker and should die first.
Right, but it's the opposite for mobile elements. So if you see a question where the older bottom leaves are dying or turning yellow first, it is a mobile element deficiency. The plant is sacrificing them. If the younger top leaves are dying first, it is an immobile element deficiency. The plant can't move the resources to them.
Spot on. Now, what do these warning symptoms actually look like? The text highlights 4 major categories of morphological changes. The first and most common is chlorosis.
Chlorosis is the loss of chlorophyll, which leads to the yellowing of leaves. It's a very common symptom, and looking at the list, it's caused by the deficiency of a wide range of elements: nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum.
Which makes sense. Nitrogen and magnesium are literally inside the chlorophyll molecule. Iron is needed to build it. If you lack any of them, the leaf loses its green pigment. The second major symptom is necrosis.
Necrosis sounds intense. It is actual tissue death, particularly leaf tissue turning brown, crispy, and dying. This is caused by a deficiency of calcium, magnesium, copper, and potassium.
Then we have the inhibition of cell division. The plant simply stops growing new cells. This is caused by a lack of nitrogen, potassium, sulfur, and molybdenum.
And finally, delayed flowering. Some plants will just refuse to flower if the concentration of nitrogen, sulfur, or molybdenum is too low.
Here is what you must remember for these lists. You have to memorize these specific groupings. Examiners will give you a multiple-choice question asking, which of the following groups of elements causes necrosis? You need to know it's K, M, N, G, K.
It seems really tricky, though, because if I'm a farmer and my plant turns yellow chlorosis, how do I know which element it is? The list of causes has 8 different elements on it.
That is the practical challenge of agriculture. You have to realize that a single symptom, like chlorosis, can be caused by the deficiency of several different elements. Furthermore, the deficiency of a single element may cause multiple different symptoms in the same plant. And to make it worse, different plants respond differently to the exact same deficiency. Identifying the true culprit requires comparing symptoms across the whole plant, looking at young versus old leaves and comparing them with standard agricultural morphological tables.
Okay, so that's deficiency. The soil is empty, too little of a good thing. But what about too much of a good thing? Let's talk about toxicity.
Toxicity usually applies to the micronutrients. Remember, they're required in trace amounts. Because they are required in such tiny amounts, there is a very narrow optimal range for the plant.
Right. A moderate decrease causes the deficiency symptoms we just talked about, but just a moderate increase causes toxicity. The plant gets poisoned.
The technical definition of toxicity here is highly specific. Any mineral ion concentration in tissues that reduces the dry weight of tissues by about 10% is considered toxic.
A 10% drop in dry weight. Got it. But this leads to my second mind-blowing realization of the episode. Because identifying toxicity is incredibly tricky. Elements don't just exist in isolation in the soil, they interact with each other. And the absolute perfect, most testable example of this is manganese toxicity.
The toxicity turf war. This is a brilliant piece of biochemistry. So if a plant is growing in soil with far too much manganese, the most prominent physical symptom you see on the leaves is the appearance of brown spots surrounded by chlorotic or yellowing veins.
The text reveals that this visual symptom is actually a biological illusion.
It really is an illusion. Here is what is actually happening at the cellular level. When manganese is present in toxic excess, it acts like a bully at a buffet line. It aggressively competes with iron and magnesium for uptake by the roots. It physically blocks them from entering.
And once it's inside the plant, the bullying continues. It competes with magnesium for binding with critical enzymes. So even if some magnesium got in, the manganese pushes it out of the way and takes its seat, rendering the enzyme useless.
And if that wasn't enough, excess manganese physically blocks the translocation of calcium to the shoot apex. It stops the structural mortar from reaching the new cells.
So the plant has plenty of manganese, but because of this microscopic turf war, it is suddenly desperately starved of iron, magnesium, and calcium.
Exactly. What visually appears as the symptoms of manganese toxicity, the brown spots and yellow veins, are actually just the induced deficiency symptoms of iron, magnesium, and calcium.
That is a brilliant trick question waiting to happen on a test. An examiner will ask, manganese toxicity induces the deficiency of which elements? And you have to know it's Fe, M, and K. The manganese isn't directly burning the plant, it's starving it of other essentials.
It perfectly illustrates how interconnected the mineral web truly is. You cannot change one variable without affecting three others.
Okay, let's take a breath and recap where we are. We know what elements the plant needs. We know what happens when it lacks them or has too much of them. But mechanically, physically, how does a plant root actually pull a dissolved mineral ion out of the dirt and get it all the way up to a leaf? Let's transition to the mechanics. Getting inside, absorption, and translocation.
To understand this mechanism, scientists studied isolated plant cells, tissues, and organs. And they discovered that the absorption of elements from the soil occurs in two distinct main phases.
Phase 1 is the initial, highly rapid uptake of ions into what the text calls the free space or outer space of the cells. This area is formally known as the apoplast.
Let's visualize the apoplast because it's a strange concept if you just think of cells as water balloons. Think of a brick building. The apoplast is the mortar between the bricks and the empty hallways between the rooms. It is the continuous network of cell walls and intercellular spaces that does not cross into the actual living, membrane-bound cytoplasm of the cells.
Because it's an open outer space, movement into the apoplast is passive. It doesn't cost the plant any cellular energy. The passive movement of ions into the apoplast usually occurs through ion channels, which are transmembrane proteins functioning as selective pores. The ions just diffuse in from the soil.
But diffusion only gets you so far. Then we have phase 2. This is the slow uptake of ions into the inner space of the cells, known as the symplast.
Using our building analogy, the symplast is the actual living space inside the rooms of the building. And all these rooms are connected by open doorways, which in plant cells are called plasmodesmata. It's the continuous network of living cytoplasm.
But getting from the apoplast the hallways into the symplast the rooms means you have to cross the locked door, the cell membrane. And the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, specifically ATP. It is an active process. The plant has to physically plump the ions against their concentration gradient.
I want to highlight the terminology here because it's used a lot. The general movement of ions in or out of a cell is called flux. When ions move inward into the cells, it's called influx. When they move outward, it's called efflux.
From a high-yield exam perspective, the distinction between phase 1 and phase 2 is constantly tested. Phase 1 equals the apoplast, which is rapid and passive. Phase 2 equals the symplast, which is slow and active. Examiners love testing which pathway burns cellular energy.
So the root actively pumps these ions into its inner symplast. But how do they get all the way up to the leaves at the top of the skyscraper?
The text confirms that mineral salts are translocated upwards through the xylem. The xylem is the massive plumbing system of the plant.
They basically hitch a ride with the ascending stream of water, and that water is being pulled up through the entire plant by transpirational pull. As water evaporates out of the stomata in the leaves, it creates a vacuum that pulls more water and the dissolved minerals with it up from the roots.
And scientists verified this pathway by actually analyzing xylem sap drawn from the stem and finding the mineral salts dissolved inside it. They also used radioisotopes of mineral elements to literally trace their path, watching the radioactive signature move from the soil into the root, up the stem, and into the leaf.
And where's this all coming from? The soil. The soil acts as a massive reservoir for all these elements. The physical weathering and chemical breakdown of solid rocks slowly enriches the soil with dissolved ions and inorganic salts.
But our sources are very careful to note that soil isn't just ground-up dead rock. It is a highly complex living ecosystem. It harbors nitrogen-fixing bacteria, other essential microbes. It holds water, supplies air to the roots, which we know they need to breathe, and acts as a physical stabilizing matrix.
Which brings us to a fascinating bottleneck in the history of life on Earth. Most of those 17 essential minerals come from weathering rocks. But there is one absolute essential element that plants cannot get from rocks. And despite it being absolutely everywhere around them, they cannot pull it from the air on their own. It is the most stubborn element in biology.
Nitrogen. This transitions us perfectly into the final and most complex part of our journey, the nitrogen cycle, Earth's ultimate bottleneck.
Let's set the stakes for nitrogen. Apart from carbon, hydrogen, and oxygen, nitrogen is the most prevalent element in living organisms. As we mentioned, it's a vital constituent of amino acids, proteins, hormones, chlorophylls, and vitamins. Plants have to fiercely compete with microbes for the extremely limited nitrogen available in the soil. Therefore, nitrogen is considered a limiting nutrient for both natural and agricultural ecosystems.
But here's the paradox that confuses so many people. The air we breathe, The atmosphere enveloping the entire planet is almost 80% nitrogen gas. Why is it a limiting nutrient if plants are literally bathing in it?
Because of its chemical structure. Atmospheric nitrogen exists as N2,2 nitrogen atoms joined by a very strong triple covalent bond, N triple bond N.
That triple bond is incredibly stable and takes a massive, almost violent amount of energy to break that bond so the individual nitrogen atoms can react and be used by living things to build proteins.
That process of cracking the triple bond and converting atmospheric nitrogen into a usable form like ammonia is termed nitrogen fixation. In pure nature, without biology, it takes extreme physical events like lightning strikes and high-energy ultraviolet radiation to provide enough sheer energy to crack the bond and convert nitrogen to nitrogen oxides like NO, NO2, and N2O.
Even human industrial processes play a role here. Combustions, massive forest fires, and automobile exhausts contribute to these atmospheric nitrogen oxides. But the biological cycling of nitrogen in the soil, how life actually passes it around, is a highly orchestrated microbial dance. Let's map this out chronologically. Think of nitrogen as a stubborn puzzle piece that only very specific bacteria have the tools to reshape.
Right, so imagine a plant dies in the forest, or an animal dies. The decomposition of the complex organic nitrogen within that dead tissue back into simple ammonia is a process called ammonification.
Some of this ammonia is volatile and re-enters the atmosphere as a gas, but most of it stays in the soil, where it's subjected to the next phase, nitrification. Soil bacteria convert the ammonia into nitrate, and this happens in two distinct sequential steps handled by entirely different microbes.
This is a massive yield area for students. You must know the exact names of the bacteria for each step of nitrification. Let's break it down.
Step 1. The ammonia, NH3, is oxidized to nitrate, NO2. Notice the ITE. This first step is handled by a specific bacteria called Nitrosomonas and/or Nitrococcus.
Step 2. That nitrite, NO2, is then further oxidized into nitrate, NO3. Notice the ATE. The second step is handled by the bacterium Nitrobacter.
Nitrosomonas and Nitrococcus take it to nitrate. Nitrobacter takes it to nitrate. The text points out a key metabolic distinction here. All of these nitrifying bacteria are chemoautotrophs.
Chemoautotrophs, meaning they don't get their energy from the sun like plants do, photoautotrophs, and they don't get it from eating sugar like we do, heterotrophs.
Yeah.
They get their biological energy by chemically oxidizing these inorganic nitrogen compounds. The chemical reaction itself feeds them.
Exactly. Now, the nitrate formed by Nitrobacter is finally ready. It is absorbed by the roots of a living plant, transported up to the leaves, and there it is reduced back to ammonia to form the amines groups of amino acids. The cycle completes.
But the soil cycle doesn't end there because there's a leak in the system. Some nitrate in the soil is lost back to the atmosphere as completely useless nitrogen gas. This reverse process is called denitrification.
And again, specific bacteria are to blame for this loss. The nitrification is carried out by bacteria called Pseudomonas and Thiobacillus.
Let me reiterate for the memory anchor because there are so many names here. The forward, helpful sequence of nitrification is ammonia to nitrite to nitrate. The first step is Nitrosomonas or Nitrococcus. The second step is Nitrobacter. And the reverse, unhelpful process of denitrification is Pseudomonas and Thiobacillus. If you can keep those bacterial names straight, you have mastered the soil nitrogen cycle.
But what if a plant doesn't want to rely on the unpredictable nitrogen cycle in the soil? What if the soil is totally dead? What if the plant wants to tap directly into that infinite 80% reservoir of nitrogen in the atmosphere?
That brings us to what I call the biological miracle, biological nitrogen fixation, the symbiotic miracle.
As we established, breaking that triple bond takes immense energy. Very few living organisms can utilize N2 directly from the air. In fact, only certain prokaryotic species bacteria have the complex molecular machinery to do it. The reduction of atmospheric nitrogen to ammonia by living organisms is called biological nitrogen fixation.
And the absolute star of this entire process is a single enzyme called nitrogenase. It is capable of nitrogen reduction, and it's present exclusively in prokaryotes. We call these microbes N2 fixers. No plant, no animal, no fungus has this enzyme, only bacteria.
These N2 fixers exist in two main lifestyles: free living in the soil or acting as symbiotic partners inside a plant. Let's look at the free-living ones first.
The sources list several. The aerobic free-living fixers, meaning they use oxygen, include Azotobacter and Bajorhynchia.
Then you have Rhodospirillum, which is a free-living fixer, but it is anaerobic. It functions without oxygen.
And finally, several cyanobacteria, which are photosynthetic bacteria like Anabaena and Nostoc, are also free-living nitrogen fixers.
But the most fascinating, complex, and agriculturally significant ones are the symbiotic fixers. specifically the highly evolved legume-bacteria relationship. We are talking about a species of rod-shaped rhizobium bacteria forming an intimate relationship with the roots of legumes, plants like alfalfa, sweet clover, sweet pea, lentils, garden peas, broad beans, and soybeans.
And it's not strictly limited to legumes. The microbe Frankia produces nitrogen-fixing nodules on the roots of non-leguminous plants, like the Alchemist tree. Now, here is a critical nuance examiners love to test regarding rhizobium and Frankia.
It's a great trap. Both rhizobium and frankia are actually free-living in the soil normally. They can survive just fine on their own in the dirt. But they can only fix atmospheric nitrogen when they are acting as symbionts inside the plant root. When they are free-living, they do not fix nitrogen. The machinery only turns on when they team up.
Okay, let's walk through how they actually team up. How do they get inside the root? The process of nodule formation. Reading this, it sounded almost like a mutually agreed upon biological hijacking.
It is a highly choreographed molecular handshake. First, the rhizobia bacteria in the soil multiply and colonize the immediate surroundings of the legume's roots. They physically attach themselves to the epidermal cells and the tiny root hair cells.
In response to chemical signals from the bacteria, the root hairs literally curl over, forming a shepherd's crook, physically trapping the bacteria. The bacteria then invade the root hair cell.
An infection thread is produced by the plant. It's essentially a microscopic tube that carries the multiplying bacteria deep into the inner cortex of the root.
Once the infection thread reaches the inner cortex, the bacteria signal the plant cells to start dividing rapidly. The cortical cells and the pericycle cells begin massive cell division. The bacteria are released from the thread into these newly dividing cells, and they differentiate into specialized nitrogen-fixing bacteroids.
Ultimately, this massive dividing cells forms a visible bump on the root, a mature nodule. And this nodule develops direct vascular connections to the host plant's xylem and phloem so they can directly exchange nutrients. The plant ships sugar to the bacteria and the bacteria ship ammonia to the plant.
If you were to walk into a soybean field, uproot a plant right before flowering, wash the dirt off the roots, and cut one of those little spherical nodules completely in half, you would notice something shocking. The central portion of the inside of the nodule is red or pink.
And this brings us to mind-blowing realization number 3, the oxygen paradox.
I am so ready for this. Why on earth is the inside of a plant root pink? Plants are green.
It is pink because of a complex compound called leguminous hemoglobin, or simply legumoglobin. It is chemically very similar to the hemoglobin that makes human blood red, and it exists in the nodule to solve a massive, potentially fatal biochemical problem.
Let's lay out the problem. The enzyme nitrogenase, which we know is a molybdenum iron protein that actually catalyzes the conversion of N2 to ammonia, is incredibly highly sensitive to molecular oxygen. If free oxygen so much as touches the active site of the nitrogenase enzyme, the enzyme is irreversibly oxidized and completely destroyed. It requires strictly anaerobic conditions to function.
But here is the massive paradox. The chemical equation for biological nitrogen fixation requires a staggering, almost unbelievable amount of cellular energy. The reaction is N2 plus 8 electrons plus 8 protons plus 16 ATP, yielding 2 molecules of ammonia, some hydrogen gas, 16 ADP, and 16 inorganic phosphates.
16 ATP for just 2 molecules of ammonia. That means 8 ATP are required for every single molecule of NH3 produced. It is one of the most energetically expensive reactions in all of biology.
Exactly. And the only way the host plant cells and the bacteria can possibly generate that massive amount of ATP is through highly efficient aerobic respiration, which requires oxygen.
So it's a biological catch-22. How do you breathe oxygen to make massive amounts of energy without letting the oxygen touch and destroy the very enzyme you're trying to power?
The brilliant evolutionary solution is the legumoglobin. The plant and the bacteria collaborate to build this pink protein. The legumoglobin acts like a highly targeted chemical oxygen scavenger. It grabs all the free oxygen diffusing into the nodule and holds onto it tightly, delivering it exactly to the respiration chain while creating a microanaerobic pocket, a totally oxygen-free zone, right exactly where the nitrogenase enzyme is operating.
It's a microscopic, pink, oxygen-free bunker inside the root. That is unbelievable. And there is a specific exception case here regarding rhizobium that you mentioned is highly tested.
Yes. Rhizobium lives purely as an aero breathing oxygen normally when it's just free living in the soil, but the moment it enters the nodule, differentiates into a bacteroid, and starts fixing nitrogen, it completely switches its lifestyle into anaerobic mode to protect the enzyme.
It's a phenomenal symbiotic trade. The plant pays the massive energy bill to 16 ATP per reaction and provides the anaerobic pink bunker. In return, the bacteria use their exclusive molecular machinery to crack the triple bond and hand over the precious ammonia.
Which brings us to our final step. The bacteria hand over this ammonia, but there is an immediate, highly dangerous problem.
At physiological pH, the normal environment inside the plant cell, the ammonia is rapidly protonated to form the ammonium ion, NH4 plus Al.
And ammonium ions are highly toxic to plant cells. They interfere with metabolism. They cannot be allowed to accumulate. The plant must instantly disarm the toxic ammonium by locking it into an organic, safe molecule. Specifically, it uses it to synthesize amino acids. It does this in two main ways.
The first way is called reductive amination. I like to think of this as boxing up a volatile package. The toxic ammonium reacts with a keto acid called alpha-ketogluteric acid. This chemical reaction requires energy in the form of NADPH, and it forms a safe, stable amino acid called glutamic acid, or glutamate. This reaction is catalyzed by the enzyme glutamate dehydrogenase.
So now the raw toxic nitrogen is safely locked away in the chemical structure of glutamic acid. The plant is safe. The second process is called transamination.
Think of glutamic acid as the mother amino acid. It contains that newly captured precious amino group. Through transamination, glutamic acid safely transfers its amino group to the keto group of a completely different keto acid, instantly creating a brand new specific amino acid.
It acts as a universal nitrogen donor, and this transfer is catalyzed by an enzyme called transaminase. By passing the amino group around, the plant uses this single method to build all the other 19 specific amino acids it needs to synthesize complex proteins.
But plants also need to transport this newly fixed nitrogen from the root nodules up to the leaves and developing seeds efficiently. They do this by forming amides, specifically the two most important ones, asparagine and glutamine.
These are formed from two standard amino acids, aspartic acid and glutamic acid. The plant simply takes these acids and adds another amino group to each, replacing the hydroxyl part of the acid with another NH2 radical.
Why go through the trouble of doing that? Why not just ship the regular amino acids up the stem?
Because of shipping efficiency. Carbon atoms are heavy, and they take up space in the molecule. Amides contain significantly more nitrogen per carbon atom than standard amino acids. They are highly dense nitrogen packages. By converting them to amides, the plant maximizes the amount of nitrogen it can transport via the xylem vessels in a single trip.
And some plants push this efficiency even further. The sources note that the nodules of some plants, like the soybean, export the fixed nitrogen as complex compounds called urides. Urides have an exceptionally, incredibly high nitrogen-to-carbon ratio, making them the most efficient transport vehicles possible for the transpiration stream.
Toxicity management, amino acid synthesis, and highly efficient vascular transport. It is a masterful, complete system from the dirt to the top leaf of the canopy.
Wow. We have covered an incredible amount of ground today, from the 1860s history of hydroponics all the way down into the inner biochemical workings of a pink root nodule. To wrap this all up, let's distill everything we just discussed into the absolute core takeaways. What are the 5 most critical facts a student must remember to survive the NEET and truly master this topic?
Number 1, the essentiality criteria. For an element to be considered essential, it must support the life cycle, be biochemically irreplaceable, and be directly involved in plant metabolism. Number 2, mobility dictates deficiency symptoms. This is the factory analogy. Mobile elements like nitrogen, potassium, and magnesium are scavenged, causing older leaves to suffer first. Immobile elements like calcium and sulfur are stuck in place, causing younger leaves to suffer first. Number 3, the manganese toxicity illusion. Excess manganese acts like a bully, masking itself by inducing the deficiency symptoms of iron, magnesium, and calcium. Number 4, the absorption pathways. The apoplast is the outer, fast, passive pathway through the cell walls. The symplast is the inner, slow, active pathway through the living cytoplasm, requiring ATP. Number 5, the oxygen paradox of nitrogenase. The enzyme requires a strictly anaerobic environment to function, which is provided by the pink oxygen scavenger, leghemoglobin, even while the host plant cell respires aerobically to provide the massive 8 ATP needed for every single ammonia molecule produced.
I love it. And now, for the 60-second sticky summary, the rapid-fire synthesis to lock it all in. Here we go. Plants are literal chemical factories superglued to the floor. They pull macronutrients and micronutrients from the soil using fast passive pathways and slow active pathways. They use these raw materials to build physical structures like the calcium mortar of cell walls and energetic enzymes like magnesium-powered chlorophyll. And when they run out of parts, their leaves tell the diagnostic story in patches of yellow and brown, ruthlessly dismantling the old leaves to save the new if the elements are mobile. And for the ultimate prize, nitrogen, they bypass the depleted soil entirely by forming a microscopic symbiotic alliance with bacteria. They create a pink, oxygen-free bunker in their roots to crack the atmospheric triple bond, safely lock the highly toxic ammonia into glutamic acid, and ultimately feed the entire food chain of the world.
Beautifully summarized. A masterclass in synthesis.
Before we go, I want to leave you, the listener, with a provocative thought. Something to mull over that builds on everything we just discussed today. We learned that the incredible nitrogenase and legumoglobin system is mostly restricted to legumes, peas, beans, alfalfa, and a few specific microbes. But imagine the future of biology. What if geneticists could transfer the highly specific genes for this exact nodule-forming, nitrogen-fixing machinery into our major cereal crops? We're talking about wheat, corn, rice, the crops that feed billions of humans. If a field of wheat could fix its own nitrogen from the air, just like a soybean plant, it would virtually eliminate the need for synthetic industrial nitrogen fertilizers worldwide. It would prevent massive agricultural chemical runoff, save billions of dollars, and fundamentally rewrite the global nitrogen cycle and the very future of human agriculture. It's not science fiction. It is the absolute next frontier of the molecular biology we just learned. Something for you to think about until our next deep dive.